Bismillahirrahmanirrahim. I am Ibrahim Akbar. My registration number is SP21BCS059 and my group members Saira Khan SP21BCS053 and Aisha Azam SP21BCS058. Today we are going to explain the topic of genetic engineering. This is the table of content of our presentation. So, what is genetic engineering? Artificially copying a piece of DNA from one organism and joining this copy of DNA into another organism. This process is known as genetic engineering. For example, tomato plant. Does not this term sounds a little weird? You might be wondering, is it a tomato or a tomato? Tomato plant is actually a combination of both tomato and tomato. Yes, you heard right. आपने बिल्कुल ठीक सुना. टोमेटो प्लांट एक ऐसा प्लांट है जो कि पोटैटो और टोमेटो के कॉम्बिनेशन से मिलकर बनता है एंड दिस इज डन बाय द टर्म ग्राफ्टिंग. ग्राफ्टिंग इज द एक्ट ऑफ प्लेसिंग अ पोर्शन ऑफ वन प्लांट इनटू और ऑन अ स्टेम, रूट or a branch of another in such a way that a union will be formed and the partner will continue to grow. The part of the combination that provide the root is called the stalk, the added piece is called the skin. ग्राफ्टिंग एक ऐसा प्रोसेस है जिसके अंदर हम लोग दो प्लांट को वर्टिकली कट करके उनको ज्वाइन कर देते हैं और उनको आपस में स्ट्रिक्टली बाउंड कर देते हैं, जिसकी वजह से उनकी जेनेटिक इंफॉर्मेशन जो है वो मिक्स होती है और एक कॉम्बिनेशन की सूरत में हमारे पास एक प्लांट ग्रो करता है. इसी तरह अगर हम जेनेटिक इंफॉर्मेशन को कट करके उनको मिला दें तो उस प्रोसेस को जेनेटिक इंफॉर्मेशन कहेंगे, जेनेटिक इंजीनियरिंग कहेंगे. सो आई होप कि आप लोगों को जेनेटिक इंजीनियरिंग का प्रोसेस अच्छी तरह से समझ आ गया. सो देयर इज अ ब्रीफ एक्सप्लेनेशन ऑफ जेनेटिक इंजीनियरिंग. It allows gene from one organism to be inserted into a cell of a different organism of different species. Examples are human gene can be inserted into a bacterium, human gene can be inserted into a cell from other animals, bacterium gene can be inserted into a plant cell. So, genetic engineering means that the DNA from one different organism can be combined. Bacteria can be engineered to produce human proteins. Human gene can be inserted into other animals. These are the prospects for the genetic engineering. There are two prospects for genetic engineering. Number one, transgenic engineering. Number two, cloning. In transgenic engineering, putting a genetic information from one type of plant or animal into another. And cloning means making genetic copies of an other existing plant or animal. So here I have a question. What are the transgenic organisms? Transgenic organism क्या होते हैं? An organism is called transgenic if it has genetic information added to it from a different type of organisms. These are the techniques for the genetic engineering. Number one, selective breeding. Number two, recombinant DNA. Number three, PCR. In selective breeding, breed only those plants or animals with desirable traits. People have been using selective breeding for so many years with the farm crops and domesticated animals. Recombinant DNA, the ability to combine the DNA of one organism with the DNA of another organism. Recombinant DNA technology was first used in the 1970s with the bacteria. PCR. PCR is a process that allows scientists to make a copy of many DNA piece of DNA. Number two, heat the DNA so it unzips. Add the complementary nitrogenous bases. Allow DNA to cool so the complementary strands can zip together. What are transgenic animals? A transgenic animal is one that has integrated a gene or a DNA sequence which has been transferred by human intervention into the genome of a cell. For example, mice used to study the human system. Chickens are more resistant to infections. Cows increase milk supply and lineage. So, there are the application of genetic engineering. Manufacturing of drugs. creation of model animal that mimic human condition and gene therapy, human growth hormones, polycyl stimulating hormones, human albumin, monoclonal antibodies, or antimetabolic factors. That's all from my side. Thank you so very much. Allah Hafiz.